Hi and welcome to our research presentation, which is about rootless hair as reliable source of forensic genetic information. So these are my team members, which is the first one is AISHAH NADJWA, the second one is DHIADINI NAQIYAH, the third one is SABRINA, and lastly is me myself, Firzana Huda. So here is the journal that we found, which is about the rootless hair being reliable source. Bear with us because there are 4 points in this presentation, which are the first one is case presentation, the second one is analysis and solution, the third one is recommendations, and lastly is conclusions.
Based on the journal Rootless hair as a reliable source of forensic genetic information, forensic DNA analysis is an important tool used in criminal investigations for human identification. Traditionally, forensic laboratories mainly use STR analysis through PCR amplification. However, this method requires DNA sample with good quality and sufficient quantity. Rootless hair shafts are commonly found at crime scenes, but they are usually ignored because the DNA inside the hair is highly fragmented and degraded. Previously, rootless hair was mainly limited to mitochondrial DNA analysis because nuclear DNA was difficult to recover using conventional forensic methods. Therefore, the researchers aimed to investigate whether advanced sequencing technologies and SNP-based analysis could recover reliable forensic genetic information from rootless hair. One of the unique features of this case is that the researchers successfully recovered both nuclear DNA and mitochondrial DNA from single rootless hair shafts. Even a few centimeters of hair produce enough genomic information for accurate forensic identification. The study also demonstrates strong identity and non-identity comparisons using SNP-based analysis and show the potential application of rootless hair in investigate genetic genealogy, cold case investigation, and identification of unknown human remains. However, several problems were identified in this case. Rootless hair contains only a small amount of fragmented DNA, causing conventional SCR analysis to fail. DNA recovery also varies between individuals and hair samples. In addition, advanced sequencing technologies require specialized laboratory equipment, expertise, and high cost. Ethical and privacy concerns related to genetic genealogy databases were also highlighted in the study. Overall, this journal demonstrates that, despite its limitation, rootless hair can become a valuable source of forensic genetic information through modern forensic genomic approaches.
In this critical assessment of forensic facts, standard forensic DNA profiling relies on STR, which stands for short tandem repeats. These require intact and long strands of DNA. In this research paper, this paper treats rootless hair like an ancient DNA, instead of trying to find long STR markers that aren't there. The research uses single-strand library SSL preparation. This technique is much more efficient at grabbing those tiny, degraded fragments that traditional forensic labs usually wash away. However, rootless hair goes through cornification, where an enzyme called Dinase 1L2 shreds the nuclear DNA into tiny bits, mostly under 100 base pairs. This makes rootless hair a dead end for standard coded skits.
Assalamualaikum and hi, my name is Sabrina ARIPIN alias ARIFIN, and I will present about the solution options. There are 5 solution options in total. The first one is use SNP-based analysis instead of STR analysis. Traditional STR analysis often fails with rootless hair because the DNA is highly fermented and too sharp for PCR amplification. The journal proposed using single nucleotide polymorphism (SNP) based sequencing, which can still recover useful genetic information from degraded DNA. This allows forensic scientists to identify or exclude suspects even when no hair root is present. Next, apply single-stranded DNA library preparation. Researchers using a single stranded DNA library preparation method optimized for degraded DNA fragments. This method improved recovery of short DNA fragments from rootless hair shaft. It also preserved native DNA fragment ends, helping with sequencing accuracy. The third one used high-throughput sequencing technology. The study used Illumina sequencing platforms such as NextSeq 550 and NovaSeq 6000 to generate millions of reads from single hair. Deep sequencing produced enough genome coverage for accurate genotype calling and identity testing. This increased the success rate of forensic investigation involving limited DNA evidence. For the fourth one, implement investigative genetic genealogy, or in short known as IgG. SNP profile generated from rootless hair can be compared with public genealogy database to identify relative of unknown individuals. IgG helps generate investigative leads in cold cases and unidentified human remain cases. The study showed genotype concordance above 99.4%, which is sufficient for relative matching. And last but not least, use advanced computational tools. The researchers developed computational tools such as IBDGEM and Astrea Impute tool for low coverage DNA analysis. These tools calculate likelihood ratios and improve genotype calling accuracy from fragmented DNA. This provides strong statistical evidence for identity or non-identity.
For our recommendation, we propose the implementation of SNP-based next-generation sequencing, or NGS, for rootless hair analysis. This is because conventional STR-PCR methods often fail when analyzing rootless hair due to the DNA being highly fragmented and present in a very small amounts. Therefore, SNP-based sequencing provide a more effective and reliable method for forensic identification. So, the first step is to collect and prepare the hair samples properly. The rootless hair is cleaned carefully to avoid contamination, and the fragmented DNA is extracted using specialized methods for degraded DNA. This step is important because the amount of DNA recovered can differ between samples and individuals. Next, the extracted DNA undergoes single-stranded DNA library preparation. Unlike conventional PCR amplification, this method is specifically designed for degraded and fragmented DNA. This allows even very short DNA fragments from rootless hair to be preserved and prepared for sequencing analysis. The third step is performing SNP-based sequencing using NGS. In this process, the fragmented DNA is sequenced to obtain SNP profiles for forensic identification. The journal showed that this method produced highly accurate genotype matching with over 99.4% concordance, proving that rootless hair can still provide a reliable forensic DNA information. Ni slide nombor 2. After SNP-based sequencing is completed, the DNA profiles from the rootless hair samples are compared with known reference samples using bioinformatic tools such as IBDGEM. IBDGEM help determine whether the DNA samples come from the same person or different individuals. The journal showed that this method produced highly accurate results even with very small amounts of fragmented DNA. Finally, the SNP results are used for forensic identification. The genetic information can help confirm identity, exclude suspects, and identify relatives through investigative genetic genealogy. This is especially useful in cold cases, unidentified human remains, and crime scenes where only rootless hair is found. Ini slide nombor 3. Here are the evidence supporting our recommendation for SNP-based NGS in rootless hair analysis. Firstly, conventional STR analysis often fails because rootless hair DNA is highly fragmented and very short. However, researchers still obtain accurate genotype results and strong identity matching using SNP-based sequencing. The study also reported over 99.4% genotype concordance, showing that SNP-based sequencing is highly accurate and reliable for forensic identification.
For the outcome of the journal, the researchers tested 80 hairs and achieved a 99.4% genotype match between the hair shaft and the donor's saliva. They successfully determined the biological sex of the donor in every case by looking at the ratio of X and Y chromosome fragments. They also generated complete mitochondrial genomes alongside the nuclear data, providing a double layer of identification.
For the conclusion, the study demonstrates that rootless hair is a reliable source of forensic genetic information despite containing highly fragmented DNA. By combining SNP-based sequencing, single-stranded library preparation, high-throughput sequencing, and advanced computational analysis, forensic investigators can successfully identify individuals from rootless hair shaft. The method achieved high genotype accuracy and strong statistical support for identity testing, even with low DNA quantities. This approach provides new opportunities for solving cold cases, identifying human remains, and improving modern forensic investigations.
So here are the journals and the references that we use to help in completing our research presentation. Thank you for listening, and we hope that you gain some information from our research presentation.